Hi, my name is Brian, and today we'll be discussing the dysregulation of endothelial colony-forming cell function by a negative feedback loop of circulating MIR 146A and 146B in cardiovascular disease patients by Chang et al. As a disclaimer, we will not be going over the entirety of the paper, only the genomic aspects of it. With the background, coronary artery disease is the leading cause of death in the United States. It is characterized by compromised endothelial or blood vessel integrity due to a bit of a plaque. Bone marrow-derived endothelial progenitor cells, or EPCs, are involved in blood vessel repair and proliferation. EPC dysregulation is highly characteristic of CAD, or coronary artery disease. It is also known that the expression of various microRNAs are critical for the function of EPCs and are clinically relevant to CAD progression. Here's a quick overview of CAD. As we see, the buildup of fatty deposits and plaque leads to blood vessel blockage. EPCs can circumvent this issue because EPCs are involved in the proliferation of new blood vessels, or angiogenesis. And these new blood vessels would be able to circumvent this blockage. So EPC dysregulation is highly associated with CAD. So the key question the authors asked is, what are the key microRNAs involved in EPC dysfunction and what genes do they affect? And the first question they answered to answer the central question is, what mRNAs are expressed differently between healthy and CAD patients? And they use small RNA sequencing and Illumina sequencing to do this. Here's a quick overview. So RNA is extracted from the disease or healthy patient cells. It's given a poly A tail and then reverse transcribed to synthesize a complementary DNA strand. Then PCR is performed, but these PCR primers have Illumina adapters on either end, the red and blue areas here. And this then creates two complementary DNA strands of Illumina adapters on either end containing our RNA sequence of interest. This DNA is then sent to Illumina for sequencing. So the adapters bind to different parts on a Illumina flow cell. The sequences undergo undergo bridge amplification, and they eventually form different clusters, which are labeled different colors. And these clusters can be read, and the sequence of DNA can be assembled from this. And then from sequence of DNA, we can derive the sequences of RNA, and we can quantify how much RNA is being expressed and what the RNA is in our disease and healthy cohorts. And in doing this, the authors identified 12 microRNAs which are upregulated in the disease EPCs and in the plasma. These are 12 miRNAs of interest. Then the author asked, what genes are downregulated between healthy and CAD patients, and which mRNAs are associated with this downregulation? If you recall, mRNAs can target specific mRNAs for degradation, thereby downregulating its expression. So the authors are trying to see which genes and which mRNAs are associated with one another. And they did this by using a microarray. So a quick overview of microarray is that they isolated cells from a patient, healthy or disease. They isolated its mRNA, then they reverse transcribed it to a labeled cDNA. Labeled cDNA is then added to a microarray. The microarray will have oligos, so different sequences which are specific to different genes of interest. If the cDNA hybridizes to the oligo, it means that that gene was being expressed in that cell. And then the authors got mRNA from healthy and disease patients to see which genes were being expressed or not expressed. So the authors identified 21 genes down here, which were significantly downregulated in CAD patients as opposed to healthy patients. These 21 genes down here, and all 21 are associated with angiogenesis, or the development of new blood vessels. They then mapped these 21 genes with expression levels of different mRNAs. So there were 12 mRNAs identified earlier, and these 6 are the 6 of the highest expressing mRNAs of the 12. And 5 of the 6 are highly associated with the downregulation of these 21 genes. So to clarify, these 5 microRNAs, their upregulation is associated with the downregulation of all 21 of these genes. This suggests that these microRNAs may bind to any one or all of these genes, and therefore mark them for degradation. So then the authors asked, which mRNAs are associated with which genes, essentially, and then they answered this using real-time qPCR. So here's a quick overview of real-time qPCR. They first isolated RNA from patients, added reverse transcriptase DNA polymerase, DNTPs, and primers into a test tube, and they basically got cDNA out of it, which then they amplified many times, and then they sequenced to see the amount of gene expression in the different cohorts. So doing that, they found that only MIR146A and 146B are significantly expressed in disease patient cells. Yeah, so as you can see here, 146A and 146B, there's significant upregulation of these microRNAs in the disease patient as opposed to the healthy patients. This 26A is one of the other 4 microRNAs described earlier, but all 4 were not significantly differently expressed in healthy or diseased patients. So next they asked, do 146A or the microRNA 146A and 146B regulate EPC function through Rho-J? I'll explain Rho-J in a sec. So what the authors did is that they use a target scan 7.0, which is a bioinformatics algorithm to predict 10 possible target genes of 146A and 146B. Out of all 10 genes, only CAV1 and Rho-J are significantly expressed in CAD patients. My bad, significantly downregulated in CAD patients, as shown here. And they found that CV1 expression is not significantly decreased when 146A and 146B are overexpressed. And they use a luciferase assay to do this, which I will not be delving into. Essentially, a way to measure expression levels. But when CAV1 expression level is not affected with 146A and 146B treatment. However, Rho-J expression is significantly decreased with the overexpression of 146A and 146B, as shown here in this luciferase assay, and in both luciferase assays. So conclusion: MIR146A and 146B are significantly upregulated in CAD patients. They are associated also with the downregulation of Rho-J. They mentioned earlier, but Rho-J has not yet been associated with heart disease, and it's involved in a plethora of other cellular functions. So these findings suggest a novel role of Rho-J in CAD pathogenesis. It also identifies 146A and 146B and Rho-J as a potential axis which can be treated to treat coronary artery disease. Some further question is, what exact cellular functions does Rho-J impact relevant to CAD is not yet known. Furthermore, there are a total of 821 differentially expressed genes which are identified between CAD and healthy patients. So what other possible interactions could exist which are not explored by this paper? And thank you so much.